Good morning to one and all. I, Sanjukta Rajans, welcome you all here this morning to the national webinar on technological intervention in plants 2020. We have with us our registrar, Dr. P.M. Patel, our distinguished speakers, Professor Ramavtar Sharma and Professor Mamta Shah, faculty members of the department, Archana ma'am, Solanki sir, Maitri sir, Modi sir, Soumya sir, and participants from all over India. I welcome you all heartily. Our program today would involve a brief inaugural session followed by technical session. We'll begin the program with our university song.
वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन गुजरात यूनिवर्सिटी आपणी गुजरात यूनिवर्सिटी आपणी विद्यातणु तपोवन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
राष्ट्र चेतना कला साधना खेल भावना विकसे
विश्व मानवी बनी गुजराती विश्व सकल માં વિલસે સ્નેહ સંપ સહકારની સાથે દેતી તું સન્મતિ સૌના સાથને સંગાથે સૌ સિદ્ધ કરે ઉન્નતિ પ્રેમ બંધુતા સમાનતાના પલ પલ પ્રસરે સ્પંદન વંદન અભિનંદન વંદન અભિનંદન वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
Now I request Archana ma'am to formally welcome the guests.
Thank you, Sanjukta, and a very good morning. It's indeed a pleasure to welcome you all here for the national webinar on technological interventions in plants. Gujarat University, with the vision and mission of our Honorable Vice Chancellor, Professor Himanshu Pandya, is on the fast track towards progress. Pandya sir has inspired not only the students, but the teaching and the administrative fraternity to excel with utmost dedication. We are very proud of the 5-star ranking by GSIRF for Gujarat University. We welcome our registrar, Dr. P.M. Patel, for always supporting us and helping us and guiding us. Thank you, sir, for joining here in time. We really are appreciating this. We heartily welcome our distinguished speakers, Professor Ramavtar Sharma, Principal Scientist from Central Arid Zone Research Institute, Jodhpur, Rajasthan, and Professor Mamta Shah, Professor and Head, Department of Pharmacognosy, LM College of Pharmacy, Ahmedabad, Gujarat, and thank them for accepting to share their research experiences with us. I also welcome my colleagues in the department, Professor Hitesh Solanki, Professor Bharat Maitre, Dr. Nainish Modi, Dr. Soumya Patel, and all the participants today. Plants have magical properties, not only to provide nutrition, but also heal. Technological interventions have strengthened the economic value of plants, thus making them better providers of nutrition and health to us. This webinar will enlighten us towards the uniqueness of plants once again. We look forward to a great learning experience. Thank you.
We request our registrar, Dr. P.M. Patel, to bless us.
Namaskar. In the occasion of this webinar, I think this is the second webinar where I joined from the department. As Honorable Madam, Botany Department told, we are a 5-star ranking and NIRF ranked in a good track university, and this is only possible through this type of webinar in the current scenario. And we can say this is the continuation of knowledge sharing. As a this is from the botany department, so of course it is of my choice also. So but as an office bearer today, I congratulate team of botany department to organize such a nice webinar where technologies can be implanted in the plants, and how the human beings can be benefited. As far as today's chief guest and honorable speakers are concerned, I personally welcome honorable sir, and madam is concerned. Namaskar, madam. I actually, fortunately, madam, in before 25 years or 20, back, madam taught us, and so I'm very much happy in the second direction also that I got my guru on webinar as a renowned guest. So I'm very much happy for that, Mamta madam, and sir. I just know that when certain plants are concerned, we still remember those knowledge sharing from her during our student life in the GU. So
It's really good that you are there and I'm seeing you at this chair.
and this is the power of teacher, and this is the power of departments of GU. So I thanks that I got opportunity to become a part of this seminar. As far as GU is concerned, we are going to develop a number of research park sections, part 1, part 2, and part 3. Our Honorable Vice Chancellor, sir, Professor Himanshu Pandya, sir, due to his vision and due to all professors and administrative staff's teamwork, we plan to go in a good ranked university where we have a collaboration with the research with the different parts like pharmacy or technological other courses and other parts of chemistry, botany, and all others. So as now in this seminar, I personally and office bearer, I congratulate the team, and once again thanks for inviting me. Thank you very much.
Thank you.
Thank you, sir.
A poster
Please.
competition was announced alongside this webinar, and we are happy that the results are ready. I request Archana ma'am to announce the results.
Thank you, Sanjukta. So, category-wise and theme-wise, I'll just announce the results of the poster competition. UG Theme A, Budding Researcher Award to Mishant Kakadia and Raj Kumar Patel. UG Theme B, Budding Researcher Award to Hemanshi Thakur, Jinal Waghela and Priyanka Parmar. PG Theme A, Young Scientist Award to Dhruva Jani and Anal Maitre. PG Theme B, Young Scientist Award to Ami Patolia and Riti Shah. Research Theme A, Young Scientist Award to Milan Wala and Dhruv Pandya. Research Theme B, Young Scientist Award to Sandhya Verma and Stacey Christian. Congratulations to all. Let us all give them a round of applause. Thank you.
Thank you, ma'am, and congratulations to the winners. I request Modi sir to thank the guests.
Thank you, Sanjukta. Respected Professor Himanshu Pandya, Dr. Jagdish Rao sir, Dr. P.M. Patel, Professor Ramavtar Sharma sir, Professor Mamta Shah, my colleagues and dear participants. On behalf of GUBS, I, Nainesh Modi, propose the formal vote of thanks. At the outset, I thank our Honorable Vice Chancellor, Professor Himanshu Pandya, for constant encouragement and guidance. I also thank Dr. Jagdish Bhavsar for his motivation and help. And I also thank our Registrar Sir, Dr. P.M. Patel, for his timely support. Our distinguished speakers, Professor Ramavatar Sharma and Professor Mamta Shah, have been very kind to share their experience with us. We are very grateful to both of you. I also express my gratitude to our judges who helped us in evaluating the posters. I thank all participants for their enthusiastic participation in today's webinar. I thank the Director Rolwala Computer Center, Gujarat University, to facilitate the live streaming of the event on University FB page. I thank Mr. Sagar Panchal from GUSAC and Mr. Dhruv Pandya GUBS for technical help. I thank Sanjukta Rajhans for anchoring this webinar so gracefully. And last but not the least, I thank my colleagues in the department for their timely help. Thank you very much. Thank you all.
Thank you, sir. Now, we shall start with the technical session. I request all the participants to post their questions in the chat box. We'll try and get back on email. The feedback form link will be active soon after the session. So, we request the participants to fill the form and receive the e-certificates. Now, we shall start with our first technical session on theme A, Technological Interventions and Plans for Better Nutrition. We have with us our distinguished speaker, Professor Ramavatar Sharma, Principal Scientist from Central Arid Zone Research Institute at Jodhpur, Rajasthan. Sir is a recipient of several fellowships and has published more than 100 research papers. He has guided many students and has developed a variety of moth bean RMO 2251 and identified a meal sterile line in Asiatic carrot. Sir has developed various molecular biology techniques like DNA isolation without liquid nitrogen, direct PCR, microwave for mutagenesis, and use of nitrogen gas for ballistic gun mediated transformation. He also has expertise on genome sequencing, differential display, molecular mapping, and tagging. Sir, we look forward to your talk on biofortification approaches and achievements. We request Professor Sharma to kindly deliver his talk.
There is something wrong.
It's fine, sir. We can wait. No problem.
No, but I shared it. Why you didn't? पूजा, can you see? कहाँ शेयरिंग में दिक्कत कर रहा है? यहाँ क्या करना है?
सर स्लाइड शो करेंगे तो आ जाएगा.
Sir, just start your presentation. You already shared your.
Slides. Hey, PPT is on.
Yes.
Yes, sir. Your PPT is visible to us.
Okay, it is visible. Okay, fine.
Yes.
Thank you.
Yeah. Just
Yes, sir.
Because there was some mistake. I'm habituated to Zoom only. So, Honorable Dr. Archana Madam, Dr. P.M. Patel, Registrar Sahib, Dr. Jagdish Ji Bhansar, Dr. Nayanesh Ji Modi, Dr. Mamta Madam, Dr. Sanjukta Rajhans, Dr. Dhruv, and other my colleagues. I am grateful to Honorable Vice Chancellor and organizers for the invitation to share my views on biofortification of crop plants. Actually, biofortification is something to address on improving or stuffing nutrients in staple crops so as to balance the diet of common man with fewer crop species available at commercial level. So we will see why this is needed and what we are missing on nutritional front. Almost half of the world population is suffering from malnutrition of iron, zinc, calcium, iodine, and selenium. However, if we see the total nutrients needed, about 40, the situation is still grave. And most of our staple crops are deficient of these minerals, at least few of them, and they don't balance the food we are eating for nutritional content. The staple crops normally world population is dependent is probably half of the population is eating the rice and another half must be eating all other crops, wheat, cassava, beans, wheat, sweet, potato, pearl millet or maize. Pearl millet and maize, it has gone out of our food table. So the best way is to concentrate the bioavailable micronutrients in these staple crops so as to reach the common man in a economic way. If you look into nutrients, the macronutrients we need in bulk, they supply us energy and building blocks like carbohydrates, proteins, fats. But some of the nutrients, and their effect is, drastic. If you don't eat sufficient carbohydrates or you don't take sufficient energy and proteins, you are weak and there will be so many debilities. But a list of nutrients is there that are needed in a very small quantity and their effect is not so obvious. And that's why it is called hidden hunger. Whereas if you don't make sufficient energy and protein, it is hunger. You are hungry. If you see the nutrients needed by plants and animals, I have just put it for the comparison because of the students only. You see, plants need only minerals, but animals or human beings are dependent on for their carbohydrate and proteins and everything on the plants. Only one carbon source, the CO2, supplies carbon to the plant, and from there it makes everything it needs in organic mineral form. But animals and human beings, they cannot. make everything from the single source of carbon, and even there are some essential amino acids and fatty acids which we don't synthesize in our system. And minerals, of course, again, we depend on the plant system or animal to animal system. Vitamins, usually we have to depend on microbes and this. Even B12, it is very difficult to get in vegetarian diet. So we have to actually diversify our diet, and nowadays we are depending on staple crops only. So if we define malnutrition as per WHO, the cellular imbalance between the supply of nutrients and energy and the body's demand for them to ensure growth, maintenance, and specific function. So our functions are not normal, our growth is not normal, and these are seen in different forms like underweight and wasting, micronutrient deficiency, and obesity is again one of the big problems nowadays because of our lifestyle. We are eating too much of energy, but not the nutrients, the minerals, and other things. The situation is grave, still grave with the children and pregnant women. Millions of people are suffering from stunting, wasting, and overweight, and chronically malnourished or acutely malnourished. So situation is really acute, and countries like India, where the income is very low, they are still developing, and we don't have a supply system, so the situation is still worst. What actually happens with the micronutrients deficiency? We have muscle wasting, low energy, weight loss, low mood, decreased quality of life, reduced mobility, infections. More infections, then because of infections, you don't assimilate more food or the nutrients, and again this becomes a vicious cycle. The confusion, mind is not working well, decreased effectiveness of anti-cancer therapy, and increased risk of hospital admissions because you are more sick when you don't have nutrition in a balanced form. If you see the deaths occurring due to various reasons, malnutrition may be sharing a very small number, but even this should not be there in the society. And what is there with the malnutrition is the secondary effect, this shaded pulse. All these diseases are there because people are malnourished. So this its indirect contribution to the death rate is tremendous, and we have to be serious about it so that our society is more happy, more contributing, and normal life serving. The causes of malnutrition, if we see, we are dependent on carbohydrate-rich food. And on the top of it, we are now tuned to take more junk food and uniform diet. We don't want to diversify, even if we can afford to have it, but we don't. Plant health is another very important issue. The plants are grown without much nutrition. We don't have fertilizers and all that applied in the soil. So plants suffer the malnutrition, and that comes to our nutrition. So we lack balanced diet. Socio-economic, sometimes society says don't eat this, don't eat that, we cannot have it, and all that. But economics is more important. We cannot afford. There is scarcity of food supply. Then population pressure, of course, and if we suffer from diseases, infectious, our assimilation is not proper. The two things which our advances in science have contributed to this is erosion of agrobiodiversity and ignorance of quality traits during selective breeding. We are dependent on only on few crops, and of those few crops only a few varieties, and those varieties are still lacking the nutritional composition. So what are the strategies to alleviate? We should really try to modify our diets and diversify it. But then there are issues, and that is most important is population pressure and economics. Then we have supplements, micronutrient supplements or food fortification we call. Without going into the system of plants, we are directly supplying some tablets and other things. So these two strategies, they are dependent supply system and our choice of the food and other things. But if we bio fortify the crops, they will intrinsically, inbuilt system they will have, and whenever you will eat rice, you will have many of those nutrients supplied to you without any extra effort and money. So there are three strategies which we follow for crop biodiversification: agronomic intervention, the supply of nutrients to the plants; plant breeding, the varieties we breed for better concentration of nutrients; then genetic engineering. If we don't have the diversity within the species of crop, then we should bring it from different other species and reconstruct the system, the pathway. So at the national level, we have some programs to handle this situation. That is Integrated Child Development Services Scheme, Midday Meal Program. They are diversifying food to give school-going children a better diet. Special Nutrition Program, National Nutritional Anemia Prophylaxis Program, National Iodine Deficiency Disorder Control Program. And every year in the September, we celebrate National Nutrition Month. This year, a fund has been created in the agriculture to improve the nutritional value of the crops, and then nutritional card of the school kids will be made so as to have the track of malnutrition and handle individual kids accordingly. And we have declared some of the high nutritive value millets and millet-like looking pseudo millets like buckwheat and amaranthus as nutritious. Just this declaration, just makes the awareness, increases the awareness in the masses for the nutrition, and at the same time it boosts the demand and allow farmers to get higher prices for these millets so that they can, if they even not replace the staple crops like rice and wheat, they enter into our food chain. Fortified foods, we know since ages, the iodine fortified salt. But now we have fortified wheat, rice, milk, oil, everything. And specifically for the three main nutrients, iron, zinc, vitamin A and D, folic acid. B12 is very important. Most of our vegetarian people are suffering from B12 deficiency, muscle pain and all that. So this fortification government initiatives are helpful. However, biofortification is still better because our supply chain may fail sometimes and it is normally not possible to reach every person. Biofortification is defined as biofortification is the process by which the nutritional quality of food crops is improved through agronomic practices, conventional plant breeding, or modern biotechnology. So we will take up one by one. Before that, we will try to sum up what actually it is. So our plant breeding depends on biodiversity. Total biodiversity, whatsoever kingdoms they have, their genes can be prospected, and through omics they may enter into transgenic approach and come to conventional breeding. But within the limits of sexual reproduction, plant species can be crossed and they can be put into conventional breeding for the improvement of nutritional level. Agronomic strategies usually target the soil physical and nutritional health. If we supply sufficient nutrition, they are reflected in the plant system and seed storage parts. And this biodiversity, the conventional breeding material and induced variations which is not yet available may make a genetic material for gene discovery through omics. Omics provides you the genes which are lacking in a plant, and by replacing that gene or introducing new gene, if the total efficiency of the plant to take up the nutrition and transfer it to the storage portions, edible portions, can be improved by these genes through transgenic approach. And once you have transferred, that variety goes into conventional breeding system. So agronomy, as I said, it just targets the soil and foliar space of the nutrients. Complete nutrition if we provide to the plant, it is more healthy, less prone to diseases, gives better yield and better nutrition in the storage part or the edible part, so as to give us better nutrition. The effectiveness or its efficiency was determined, and for the zinc and selenium especially, and scientists found that yes, if we supply zinc and selenium in the soil, plants take up several folds, 5 to 100 folds more selenium in the seed, and that is available to human beings in turn, and our nutritional deficiency can be taken care of. However, breeding nutrient efficient varieties has been considered sustainable. Availability of fertilizers is again a question. Normally in developing countries we don't have sufficient fertilizer source for their independence to fertilizer application, and this system is independent of fertilizer application. It increases the efficiency of plants to mobilize the immobilized nutrients and absorb it and transport to the seed or edible parts. Another thing which is wonderful this year is United Nations has declared this year as Year of Plant Health, and this is very good to create awareness among people that how plant health is so important. And of course, we depend for everything on the plants, but if plants are healthy, they are full of nutrition, they will end our hunger and malnutrition also, along with the poverty, and they have impact on everything. Every socio-economic front plants have their bearing. Selective plant breeding is just what we call conventional plant breeding. So there are the issues which we have. Either it could be transgenic also, higher capacity to accumulate micronutrients. This depends on genes. Increase their bioavailability. How to increase bioavailability is there are certain inhibitors, anti-nutrient factors in the plants, and there are some promoter substances in the plants. So we can promote the promoters and just reduce the quantity of anti-nutrients so as to make them more assimilable in the gut. A list of enhancers and anti-nutrients or the promoters. You can see phytic acid, fibers, tannins and polyphenols, oxalic acid, hemagglutinin, goitrogens, toxic heavy metals, whereas promoters are something like organic acid, phytoferritin. This makes bioavailable the iron, some free amino acids, long-chain fatty acids, insulin, and this promotes calcium bioavailability, and all those things, riboflavin, selenium promoters. So, when we were breeding or improving the crops, our targets were to increase crop yields, enhance crop resistance to pests and diseases, and tolerance to abiotic stresses such as drought and salinity. However, while we were doing this, we were ignoring the quality aspect of the food. And ultimately, the varieties started losing those traits. And presently, most of the varieties are low in important micronutrients like Fe, zinc, copper, vitamins, and other things. Plant breeding starts with the available genetic variability. If we don't have variability for the content or the concentration of minerals and other things, other important nutrients in the germplasm, we cannot have any advancement in our breeding effort. But if there is diversity, we have to screen it, identify the genotypes that are desirable for different minerals, or there can be some genotypes which are good at several minerals in one. Sometimes we have to bring the variability from wild relatives and land races. And that is one example for wheat. So what do we identify is, we identify the parental genotypes, parents from the available diversity in the germplasm. This allows us genetic studies, molecular marker development, and parent building for improvement of the crops. Or the existing varieties or crops, if they have higher quantity of some nutrients, we can directly release those varieties in the system without much research. When diversity was studied for minerals and vitamins in the edible tissues and bioavailability in the human guts, there was good diversity available in rice for Fe gene, in the beans and peas for same Fe and gene. Wheat selenium content was transferred from distant wild relative. However, tuber and fruits, they suffer from variation. So they have little less variability and usually may need interventions from genetic engineering. Many international programs are now working for nutritional enrichment. Among them, Harvest Plus is one. Harvest Plus has few crops in its study: rice, beans, cassava, maize, sweet potato, because they are staple crops. And major targeted iron, zinc, and vitamin A. But now they are including several other micronutrients in their studies and other crops also. So they have already bred 300 biofortified crop varieties, and they are doing well in Asian and African countries. They have handled much of the malnutrition. As far as National Agriculture Research System is concerned, which includes ICAR and the agriculture universities, we have total around 5,000 varieties released. But the emphasis on nutrition is recent. Still, we have several varieties released for this purpose also. Rice for zinc and protein, wheat, iron, zinc and protein, lysine, maize again lysine, tryptophan and provitamin, and there are several varieties of maize written on the lower side of the table. So increment is quite conspicuous, good well, and probably we will be in a position to handle our malnutrition conditions. Pearl millet, lentil, cauliflower, we are bred for beta carotene, sweet potato for anthocyanin, vitamin C in pomegranate, in addition to iron and zinc. Mustard was bred for against anti-nutritional factors like glucosinolate and erucic acid, and soybean for trypsin inhibitor, so that the protein digestion would improve. There is one example from Gujarat farmer, Vallabhbhai Varsamabhai Marwania. He bred one variety of carrot, gajar, Madhuban gajar, which is high in beta carotene and iron. He was conferred with National Award and then Padma Shri Award. So I congratulate Gujaratis. Molecular breeding. Whenever we go for conventional breeding, we have to phenotype all the plants for the nutritional concentration, but it becomes very expensive and time consuming. However, if we have molecular markers attached to those traits, it becomes a little simpler. And the trait is actually decoded in the form of Mendelian genes, because those markers can be handled in a Mendelian fashion. And the trait ultimately comes with it. And for that purpose, we need different populations. We use different populations like F2 that are segregating, Pacros, RILs, NILs, association mapping payments. Earlier, we used to have RAPD, SSR, and FLP markers, and genotyping was needed of the segregating population and looked for co-segregating markers. Nowadays, SSR and SNPs are more rampantly available, and our strategy has shifted to whole genome sequencing. It is quite cheap, fast, accurate, precise. How they have the markers, a single marker is responsible for iron and zinc, and there are this another position, but then there are individual markers for iron and zinc. The most important, or having the locus having higher impact, can be used for transfer of or tracking of the trait. This was an example from pearl millet. It was successful, and varieties are there. This is for citrus fruit, and it was improved for beta carotene. If you see the two parents, they had variation. But if you see the progenies, they are having still more variation, minimum and maximum, 0.0, 3 for phytoin, and maximum 0.71, which is higher in both the sides to the parents. This is called transgressive segregation, and such transgressive segregations are very useful in plant breeding. And then these tags, the locus were available with the markers. So now you can track the trait, the nutritional component through these markers and incorporate in the variety you desire, the elite variety. And there are several markers available for Fe and zinc in rice, wheat again Fe and zinc, manganese also, then barley, then again maize grain Fe, zinc, beans, soybean calcium, and oilseed rape. I can't see it. Zinc, normally it is zinc and iron. Then oilseed rape we since have seed Fe, but oleracea we need leaf vegetable. So it is high in calcium and magnesium, high in leaf. So we have markers for all these, and breeding efforts are going on. Several varieties have been bred, several are in the pipeline. Transgenic approach is little different than what we do in the conventional breeding. There we are using well-established varieties with cross-make combinations. So the environmentalists normally don't question it. But as soon as we shift to transgenics, there are many issues associated with this, the safety and bioregulations. Still, research is going on, and at several times, it becomes imperative to adopt transgenic approaches. It is, of course, rapid in the sense if you breed through conventional methods, it takes several years to cross, follow the segregating population, then make it homozygous again, release it, and all that. 15 years, 10 to 15 years. Now there are some fast methods, but still you need more efforts. Here, since you are using single gene with very good promoters and everything for its expression, it is put into the variety, elite variety, and the result is there, immediate, and you can go for releasing everything. But because of the issues I talked, they are not normally preferred. Then, however, it is easy, but it needs big research effort prior to adopting it. You need to have genes. You have to identify the genes that are really limiting the absorption or translocation or storage in the seed. And you have to replace that particular gene in the plant, elite plant variety, so as to improve the malnutrition or so as to improve the nutritional concentration. Several genes can also be introduced simultaneously, just like in our conventional plant breeding. Gene editing is another good method where we are not introducing varieties with the constructs and all that, but we are replacing the gene on homology basis, should be more acceptable and more rewarding. And this is only option available when breeding fails because of no diversity, crossability issues, and absence of sexual cycles. Some of the plants they don't come into sexual cycles. Target genes are to be known for uptake, transport, storage, bioavailability. Bioavailability in the gut, and it directly has the bearing with inhibitors and promoters. So this to know all these things is really time consuming, demanding, and need a lot of money. And of course, you have to have big breeding populations or segregating populations to actually know or decipher the gene function. It all depends on the genetic diversity and genetic material available. And only then you can really target a gene or identify a gene to be used for our purpose. In order to establish functional relations among the genes, nowadays high throughput methods are available. Genome sequencing has become very cheap, and now we are analyzing the genome sequences, the transcripts, the proteins, metabolites, and nutrients, and establishing functional relationship among them so as to identify the genes of interest. Omics has several dimensions, several instrumentations involved. All this study should be put together in systems biology, and we have to come out with certain target genes. There are certain genes known today, and a lot of research is going on. There are genes for uptake of nutrients from the soil. Their efficiency can be increased. Phytosidrophore is known to us for some time. Then from in the roots itself, after absorption, the nutrients have to be loaded into xylem. Once it is loaded in the xylem, it has to cross the barrier of root and shoot. There the genes have. Then they are to be distributed among leaves, stem, and then seed. Again, genes undergo. Then phloem loading for the seed, and then loading in the seed. Every step is under the control of genes, and we have genes. Some of them are common in several steps. This is not only sufficient. Then remobilization is very important in plants. When they go under senescence and towards end of their life, they just take out the nutrients available in their leaves and stem and just remobilize it towards the seed. Nitrogen is mostly remobilized from leaves to the seed in most of the crops. One of the best examples is golden rice. It is a classical example where the genes were not available in the rice seed to make beta carotene. They were taken from daffodil, but daffodil genes were having two steps from phytoin to lycopene, and that was replaced by a bacterial single gene. From phytoin to lycopene in a single step, the process takes place, and it is really shorter. So it is a good example of reconstruction of the pathway to enrich the rice seed in beta carotene, which is called golden rice, yellowish in color. Wonderful. Transgenic banana is another good example. Already there was banana in Papua New Guinea, which was orange in color and found to be high in beta carotene. But the crossability and those were the issues, and conventional breeding was not found suitable. So the phytosynthetase 2A gene was isolated and put into uh, the elite variety, and it was bred. It was transformed to give higher beta carotene. Another is super banana, where the genes were taken from maize and bacteria. Two genes were put into the dwarf Cavendish variety and a new good variety with high yield and high beta carotene was produced. These are the pictures to tell you the story. There are several crops now have been made transgenic through agrobacterium, vitamin A, rice. The genes they have put is phytoin normally from daffodil, phytoin desaturase gene from Erwinia, the bacteria, the phytoase gene from maize, daffodil, and from any species you can take and put into any variety. Phagorous ferritin for iron, ferritin, zinc, soybean ferritin, and aspergillus. This is fungus gene, flavus phytase gene. So we have several examples in wheat. Soybean ferritin was used. Maize again, cassava, canola. Canola is nothing but a mustard variety devoid of glucosinolate and erucic acid. There is no transgenics released yet because of regulatory associated regions. Still, we need to ensure that there is no interaction with other nutrients. Potential allergic responses we have to determine. If there is any alteration in plant growth or anything, stress responses, we have to drop it. Omics studies of transgenics was done and no safety concern has been issued. So probably sometimes we will start accepting it. And people, scientists have said that the metabolic changes that occur at RNA or protein level are higher in conventionally bred varieties than the transgenic varieties. In order to address the safety issues, we can go for CRISPR-Cas or some other editing tools just to replace the genes, available genes, not to put any other new gene. And there is one wheat variety bred by UK's John Ennis Institute. Benefits are obvious of biofortification, total 3 procedures. The food reaches to most vulnerable people, that is nutritionally rich in a routine way, inbuilt system way. Increases the nutritional level of staple crops and we service the human beings. It is most effective since recurrent costs are not involved, and this same germplasm can be shared with many countries, can be used to breed additional varieties. It is sustainable because no recurrent costs are involved and no government supply system is needed. So that is wonderful system if helps to introduce all the nutrition required. There is wonderful result it has given in African countries, even in India, Maharashtra. So beta carotene, sweet potato, it has increased vitamin A and the deficiency has been reduced. Iron has been increased. People have more iron for the blood and better growth, lysine rich QPS, quality protein maize. This quality is for amino acids. So they supply the limiting amino acids or essential amino acids, especially lysine and tryptophan. However, still I recommend diversification of food. Commercialization of agriculture actually eroded biodiversity. So if we again go to promote traditional agriculture, could be better, but with a thinking over economics also. Rooftop and terrace kitchen gardening may help improve mental and physical health. Biofortification could be option to resolve the issue of hidden hunger taken systematically. Genetic engineering should only be adopted where molecular breeding or traditional breeding fails. Thank you very much. Thank you.
Thank you so much, sir.
Yeah. I think I'm in time.
Yes, perfect, sir. Perfect.
Yes,
Sanjukta?
ma'am. Yes, ma'am. The second technical session is on theme B, technological interventions in plants for better health. We have with us our second distinguished speaker, Professor Mamta Shah, Professor and Head, Department of Pharmacognosy, LM College of Pharmacy, Ahmedabad, Gujarat. Her area of research is development of physico-chemical quality parameters for single plant as well as their formulations, isolation, and identification of chemical markers and development of analytical methods using HPTLC, HPLC, HPLC-MS, GC, GCMS, etc. Mamta ma'am has several publications in national and international journals in the field of herbal drug research. She has received several awards for best presentations and research papers. She has completed several projects, has guided many students, and has wide collaborations not only in India but abroad as well. Ma'am, we welcome you here and look forward to your talk on technological interventions in plants drug research. We request Mamta ma'am to kindly deliver her talk. Mamta ma'am?
Yeah, can you hear me?
Yes, ma'am, you can start.
Yeah.
Yes, ma'am.
Shall I share my screen?
Please.
Shall I start?
Yes,
Just
yes,
a
ma'am.
minute, huh?
Yes, ma'am.
Is it visible?
Ma'am, please share your screen.
It is not showing my screen.
We can't. No, ma'am, not
I
yet.
have to do that.
Sanjukta,
ma'am.
is Sagar there? So can you ask him, request him to
Yes,
help,
ma'am. Yes,
ma'am?
ma'am.
Please.
Ma'am, you can see the share screen button below, right? Ma'am just left the screen.
No issue. Such kind of things happens. So, as this is not, we all are not comfortable with this new technology, and particularly senior people, because they are very good in just delivering lectures. If you are in front of him, just they will mesmerize you. But as these things are newer for them, so it happens. See, she is back again.
I don't know what is wrong. Something I'm not able to see the screen. I mean, I'm not seeing the screen here. Let me see.
Ma'am is audible actually.
Ma'am, is
Yes.
there a problem with the internet? So I have a cue from my technical support that if you want, you can keep the video off and you can present. Maybe if there's a problem with the signals, we can do it that way.
Let me see.
Then maybe we can see the presentation.
Maybe. I stop my camera.
Yeah, I don't know. Yeah, Sanjukta, I just asked Sagar.
Sagar said yes.
Stop my camera. I'm stopping. I'm trying to. Shall I? Let you a second. Somebody from present.
Ma'am, you are not audible
Madam?
properly. We are not getting your voice continuously.
What I'm, if I share presentation to one of you,
Okay, I got it.
you can.
Sanjukta, ma'am, Sanjukta,
Yes, sir.
ma'am wants to say if she shares presentations to us, anyone from us can just share that presentation on screen and scroll on the slide as her suggestion.
Okay, so Sagar has just sent an email ID. So ma'am, can you just share it on that email ID? Here you can see it in the chat here. They will present it.
Open. I am mailing you. Madam,
Okay, yeah.
I'm mailing you.
Okay. Okay, ma'am. So she will, Mamta ma'am will share Archana ma'am. Okay, ma'am.
Okay.
It is
Then you share it with Sagar.
Yeah, sure.
Problem today. Yesterday it went very well.
No issue, ma'am. Don't worry.
No, don't worry, ma'am. It's fine.
This is PowerPoint presentation. It takes a bit time.
Some issues in connectivity only.
Actually, 20. It's not showing today, but I don't know what is the reason. It's taking time also. Yeah, it has come. Shayad aaya hai. Is it there?
Yeah.
Is it there, madam?
Yeah.
Yes, it is uploading.
It is coming.
Yeah, it's coming.
Yeah.
No. The same thing which was happened earlier.
Ma'am, make it a slideshow.
Because
Ma'am, just start your presentation.
attachment
We'll
also
able
is
to see it.
taking
Ma'am, just start your presentation. We'll able to see it.
time.
Click on the slide. PowerPoint at lower.
Just use मैम आपका माउस यूज कीजिए
Click on the
और
PowerPoint.
वो जो नीचे स्लाइड पावर पॉइंट दिखा रहा है ना उसके ऊपर क्लिक कर दीजिए. यस सर.
This is what I did.
सेम थिंग. आपके लेक्चर में शुरू में ऐसा ही हुआ था ना सर. देन यू
So when I
yes,
found
sir.
if I
आपने वो
click on
क्लिक
that,
कर दिया
it
इमीडिएटली
starts.
स्टार्ट हो गया था. मैम कैन यू हियर मी? I think she left again.
Yeah, I think she is trying to connect, so we have to wait.
Yes, ma'am. She just left the studio. She's trying to rejoin once again.
Ma'am, can you start that share screen from your side if you have received this?
No, she has not sent me the email as yet. I don't have it.
Okay.
Otherwise, I
Okay.
would.
Yeah.
So I guess, Sagar, maybe we can see the Gujarat University video till then.
Yes.
I'll try to call her, but I don't know.
Sure, ma'am. Sure.
Yeah.
Sure. Sagar, just
Located in the heart of UNESCO World Heritage City, Ahmedabad, Gujarat University with more than 4 lakh national and international students makes it a large and inspiring place to work and study. 270 acres of lush green eco-friendly campus in conjugation with new knowledge and ideas. University meets societal challenges through strategic investments in education, research, creativity, and entrepreneurship. Extremely modern and state-of-the-art infrastructure with International Convention Center, Olympic Standard Sports Complex, open-air theatre, and Genetic Diagnostic Center. Foresight into technological life cycle and agile systems takes Gujarat University far ahead of other higher educational institutes. Promotion of research, consultancy and extension for benefit of society through Dr. APJ Abdul Kalam Center for Extension, Research and Innovation. Strong research and unique study programs including 2 finishing schools and research park bolster the industry-academia collaborations and also allure national and international students and researchers. Only state-funded university to incorporate 3 nationally funded incubators, which play a pivotal role in shaping and nurturing the startup ecosystem in Gujarat. Incorporating a strong sense of openness, Gujarat University actively participates in public debates and dialogues, cross-cultural university partnership through national and international collaborations to enhance global competitiveness. 3 world records and 2 national awards, together with history of individual and institutional level achievements, makes Gujarat University committed to excellence, innovation, and highest standards in higher education. The NIRF 2020 ranking places Gujarat University at the 44th position nationally and 12th position among states-funded universities in India. Gujarat University offers a rare combination of elite academic performance and integrated learning environment, thus making Gujarat University a sought-after institution, which has proved its ingrained motto of Yogaha Karmasu Kaushalam.
So, Jugda, have you kept the message on the FB chat and the YouTube chat?
Yes, ma'am.
Okay.
And still I'm having, I think, problem in sharing. I don't know what is wrong, but it is not getting shared. The same screen is, I mean, okay. हाँ. थયું ક્યારે શેર તમારે? Sorry for the trouble, but I don't know what is wrong today.
Actually,
हाँ हाँ हमारी लाइट कर हमारे लाइट की सेटिंग करो ऊपर से.
you,
It's fine now.
after sharing, you click on the PowerPoint
हाँ.
slide.
છે તો હું
It
કરું
will
ચાલુ
come.
અહીંયાથી.
ના તમે જ કરો મેડમ.
You start sharing.
ફર્સ્ટ ઓફ
Okay.
ઓલ જો નીચે શેર સ્ક્રીન હશે, શેર સ્ક્રીન લોઅર સાઈડ ઉપર સ્ક્રીનમાં.
Share screen. You click on that.
મમતા મેમ
हाँ बोलो.
તમારા સ્ક્રીનમાં નીચે એક શેર સ્ક્રીન કરીને હશે જો.
हाँ છે
Mute.
પણ ત્યાંથી
હા.
કરું છું ત્યાંથી કરું છું બટ દેન એ
Okay.
આ પ્રેઝન્ટેશન નથી હોતું તમારી આપણી સ્ટ્રીમયાર્ડની સ્ક્રીન જ લે છે there are two screens open right પણ આ સ્ક્રીન લેતું નથી એવું છે why I don't know તો રોજનું છે અમારે actually we do online teaching only but I don't
Okay.
know what is wrong today.
યા યા
Ma'am,
Let me this one more time and if I'm able to I'll share all or else મેં મોકલ્યું છે મેડમ તમે ત્યાંથી કોક પાસે શેર કરાવો so that would
ઓકે
be
ઓકે હા હા હા.
let me see
Sanjukta,
one more time.
yeah ma'am, you try one more time, otherwise I will ask these people, technical
Yeah,
people,
yeah.
to just share it. Sanjukta, till then you also try.
Yes, sir. Yes, sir. We are trying, sir. We are trying from here.
Okay.
થયું મેં કર્યું છે એથી try but it is StreamYard screen only which is
મેમ હવે
Getting
તમે તમારે
shared.
પીપીટી નહીં મેમ તમે શેર કરો પછી તમને ભલે સ્ક્રીનયાર્ડ સ્ક્રીન દેખાય યુ જસ્ટ સ્ટાર્ટ શેરિંગ એન્ડ સ્ટાર્ટ પીપીટી તો અમે લોકો અહીંયા જોઈ શકીશું.
ઓકે તમે ત્યાંથી કરો છો તો પછી હું આઈ એક વખત પછી ટ્રાય કરી લઉં મારી પાસે બીજી પેનડ્રાઈવમાં છે બીજા લોકેશન પરથી રાઈટ તો થતું હોય તો જોઈ લે પછી ની ટ્રાય સ્ટાર્ટ
મેમ હું અહીંયાથી શેર કરી દઉં જસ્ટ મિનિટ મને પ્રેઝન્ટેશન મળી ગયું છે જસ્ટ
કરો છો તમે ત્યાંથી થઈ ગયું તમારાથી
Yes, yes,
યસ મેમ યસ મેમ
yes ma'am, he will
So I
he
can
will
start
share.
from here.
Just wait for a moment.
Shall I start?
Wait.
So,
Yes,
yeah.
yes, yes.
So I apologize for all the technical hitches, and we are now back with Mamta ma'am's lecture. So requesting all to stand by, and Mamta ma'am, your presentation
Yeah.
is here. So please
Yeah.
start.
So
Thank you.
it has been shared, right?
Yeah, please.
Everybody is able to see that clearly.
Yes,
Yes,
yes.
ma'am.
Yeah.
And
Then I shall
whenever
be
you say
starting.
next, so whenever you
Yeah,
say
yeah.
next, we'll.
I shall be confirmly telling. Good afternoon to one and all. I'm sorry for the disturbance. It's a technical error which occurred. I don't know what happened, but then મોડા તો મોડા પણ થયું તો ખરું ને like see we all know that there is a huge increase in the interest in the use as well as the research in the medicinal plant for the treatment of variety of treatments, ailments. And if we see this present COVID era, you will see every other day there would be an advertise from Ayush department or from any other, some other government department which are trying to promote use as well as research of these medicines, that is medicinal plant, to obtain an either known, kind of a chemical moiety, or maybe with unknown kind of chemical moiety with new use or newer or better potency and less toxicity. So for this thing, there is actually a need to understand and implicate modern science to uphold the significance of these medicinal plants, right? So this is some information regarding contribution of the medicinal plants towards the drug discovery. If we see the literature, there are approximately 80% of antimicrobial, cardiovascular, and immunosuppressive as well as anti-cancer drugs which are there, which are currently being prescribed in routine for the treatment of related disorders. And research has led to the collection of remarkably diverse array of chemical compounds, and the number is more than, I think, 1,40,000. And if we see natural, why we go to nature for seeking the help to get a new molecule with a potential activity as well as more safety and stability, because natural molecules offer more drug-like features than those from combinatorial chemistry in terms of functional groups, chirality, and structural complexity. Next, please. Mine has gone. So the next slide shows the chart, which is showing you the actual presentation where the drugs which are derived naturally are showing more resemblance to the actual drug like property, and those drugs which are derived from synthetic source are not that closer to the drug like property, that is in chirality and functional groups. So always natural products are obtained for as a better source for getting a drug like molecule. Next, please. So this is a forest chart and it shows the presentation. And this is the next slide, shows that what kind of contribution is there from by the natural products or medicinal plants. We can see here in this pie chart that about 64% of these drugs, 64 drugs are directly unaltered natural products, and that comprises to about 4% of the total therapeutic molecules available in the market. Then about 141 drugs are actually botanical mixtures, and that constitutes about 9.1%. 320 drugs are natural product derived, that is derivatives. So basically they are used as a starting material and then they are made in the laboratory. And about 61 drugs, that is about 4% again, are synthetic molecules, but they are basically the basic pharmacophore is from the natural source. Next is, these are some examples which I have tried to quote here, wherein we can see that different sources are there of different natural sources are there, like medicinal plants, marine source, biopharmaceuticals, antibiotics. All these examples which are quoted here, there is Taxol, artemisinin kind of compounds. They are all very powerful clinically used anti-cancer agents. Marine source, I have quoted some examples which are of importance, like Prialt is 1,000 times more potent analgesic than morphine. So you can see the extent of potential which is lying in this kind of sources, that is natural sources. And we know that there are a number of biopharmaceuticals also and antibiotics also in the market which are currently being used for the treatment of variety of disorders. Then these are not only therapeutically active compounds, but also consumables like excipients which are used in making the various dosage forms. Then nutraceuticals, spices, herbal cosmetics, these are all obtained from the natural source. And even drugs of abuse like hallucinogens, carcinogens like cannabis, opium, these are all obtained from the natural source. So we are gifted a very good source by nature, and we can always opt for getting new kind of molecules with more potency and safety. So, as I told you, there is a huge increase in the research and interest. I mean, there is interest in research world over, and this is a graph which I have tried to show here, which shows that there is a steep increase in the field of medicinal plant research, and this is a publication trend which is shown here from 1940s to till date, and you can see there is a huge increase. The latest one shows about 50,000 publication a year in different fields of medicinal plants. And these are the fields of interest as far as medicinal plant research is concerned. This is the pie chart which shows presentation, of different kinds of publication by discipline from 2008 to 2018. You can see there is about 18%. There are about 18% publications which are of plant science field. Then there are 15% publication which is from the field of molecular biology. It's a very upsurging field. Then there are about 13% publication from the agricultural field, and if you see pharmacology, toxicology, chemistry, then it makes more than about 40%. And this overall shows that there are number of fields which one can enter into to go for drug discovery from the natural source. And so we can say that natural products have played and will continue to play a key role in drug discovery, and are therefore traditionally claimed as cornerstones of the drug discovery and development. Now, how this medicinal plants contribute towards this drug discovery? They can be a direct source, as we have seen, like atropine, cardiac glycoside like digoxin, and many more compounds like Taxol, camptothecin. These are all directly obtained therapeutic agents from natural source. So they can serve as direct source of therapeutic agents, like as both pure drugs as well as phytomedicines. They can serve as a precursor for semi-synthetic chemical compounds, or designing of new analogs with either improved therapeutic activity or reduced toxicity. There are a large number of examples here also in this category. Then they can be source of prototypes for design of lead molecules. One can get always idea, and then one can modify the structure and get the new molecule of better potency. Then they can be, these medicinal plants would be serving as a direct raw material for herbal drug industry as well as Ayurvedic industry and other traditional medicinal systems. And they can be source of standardized extracts, and these standardized extracts can serve as a raw material for herbal-based dosage forms for the industry of nutraceuticals, cosmetics, herbal supplements, and traditional formulations. So there is a huge kind of you can see possibilities and areas which one can go for this drug discovery from natural source, specifically from the plant drugs. Now, if we see the technological interventions in the plant research, one can basically divide this approach into two categories. One is random approach, and the other one is guided approach. And if we see random approach, here nothing, one goes randomly for the selection and collection of the drug material, starting material to go for the discovery of the drug molecule. And there would not be consideration of any information regarding the selection as well as collection of this basic plant material. And that is normally, like if I want to go for, I'll be going for systematically, like I'll go for the traditional literature, or I'll go for the ethnobotanical or ethnopharmacological literature. But here, in case of random collection and selection of the plant material, directly a plant material which is available, it is selected, and it is screened for the activity as well as phytochemistry. Now, here this approach was taken over by CDRI 3 decades ago, and they screened about 2,000 plants, and they could not get any new chemical entity, and the entire process was a failure. But NCI, that is National Cancer Institute of USA, they studied this about 35,000 plant samples from about 12,000 species for anti-cancer activity, and this they had taken up between late 60s to mid 80s. And during this period, they screened about this many number of plant species, that is 12,000 plant species, and they ended up with two blockbuster drugs, that is paclitaxel and camptothecin. We all know that both these drug molecules, they are used clinically for the treatment of variety of tumors. And second approach is a guided approach where it can be categorized into three levels, there are 3 stages: pre-drug stage, quasi-drug stage, and full drug stage. Now, this is actually a systematic approach where one has to take care of every step one takes up. So it starts from the selection of the plant material to the final stage, final stage of the full drug development. So first is the pre-drug stage, that is information-driven approach for the selection of medicinal plants. And then there's a quasi-drug stage where the drug discovery is actually starting. After the selection, we subject that plant material to the drug discovery process, and that starts with phytochemical screening, biological evaluation, and then dosage form design. So that is the quasi-drug stage. And then the third stage is full drug stage, where here what we do is we develop all the aspects of safety and stability of this molecule, whatever we are getting, and then we try to go for the synthetic analog of this natural lead, which is identified, to get a better potent, better drug with better potency and less toxicity. Or we can directly use this molecule and combine with synthetic molecule to get again a new lead. And also we study the metabolomics approach, that is we try to see what all are the metabolites of these molecules, which are, that is pharmacodynamic and pharmacokinetic studies are done. So first I'll be going for each of the stages systematically. So for pre-drug stage, we target plant selection. Now this selection of the plant is actually the cornerstone of research. Why? Because if we go wrong in this particular stage, which is an initial or first stage of our drug discovery, then what will happen is we will not be getting the desired or whatever we are trying to get result of our kind, like whatever we are targeting. So such selections are to be done on the basis of the information from the varied sources, like traditional knowledge, ethnopharmacological approach, chemosystematic approach, ecological interactions, and computational approach. These are different approaches, and conventionally when we see traditional knowledge approach, ethnopharmacological approach, and chemosystematic approach along with ecological interactions, these are all conventional approach. The addition into this, the technological intervention is there with the entry of computational approach. But here also this in case of traditional knowledge, what we earlier were doing was we used to see the books of traditional knowledge, that is Ayurveda, Siddha, Unani, or maybe traditional Chinese medicine. We go through the books and we'll try to search for the probable plant which will be having, like if I am targeting some screening for anti-inflammatory or anti-cancer plant, then I'll go for the literature survey using this material, recorded documents of ancient time, and I'll try to surf through those, and I'll try to get the information about these plants having these particular activities, and then I'll try to enlist them, and then I'll start my search. This approach is actually very good approach, is now facilitated in this present era with the help of computational documentation, right? So here we don't have to go through all the literature, hard books or hard copies. We have now information in the form of digital libraries, like traditional knowledge digital library. This is TKDL, commonly known as. That is compilation of about you can say 1200 Ayurveda, Siddha, Unani formulations, and it has access to about 2,500,000 medicinal formulations also. So you can see we have digital library which you can search for your purpose very easily. So this is the technical advantage of technology which we have. And likewise, there are a number of other databases which are based on traditional knowledge. And these are examples quoted here. Next, please. The traditional Chinese medicine database @ Taiwan. This is also known as TCM database @ Taiwan. And this is again a freely available database which can be used for surfing. It contains about 60,000 TCM compounds from over 450 herbs, minerals, and animal products. And it provides comprehensive ingredient to TCM mapping combined with 3D structures of each ingredient. So see what kind of content this database have got. And then next is TCMID database. Again, this is very good database which has got huge number of entries into that for treatment, data on drugs, targets as well as diseases. And so we can use, rather than going to traditional books and surfing through them, it would be taking long time. It would be very tough also. We have at our end now this kind of databases which are available digitally, and we can have our desired plant selection. Next, please. Then similarly for ethnopharmacology, earlier people used to go and do surveys and then document the things, and from that documented literature they used to select the plant and start the work on screening and biological and phytochemical screening. But these days again this has been facilitated by available databases like NEPA Alert, Lady Oak College.edu database, Phytochem database, Herbamet database. Large number of other databases are available which can be directly seeked for to select for the selection of the plant material. Next, please. These are some examples. Successful stories are there, which the researchers they use this ethnopharmacological data to get the successful drugs like Gallegin, which is a template for the synthesis of metformin, very popular drug which is used for the treatment of diabetes, and artemisinin, we all know, a latest entry into anti-malarial drug. Both of them were discovered on the criteria of ethnopharmacological data. Next, please. Chemosystematic approach. Here, we know that, being a botanist, we know that family-wise there would be a concentration or distribution of secondary metabolites in the plant kingdom, and that serves as a signal that if this family is there, like Solanaceae family is there, then there is a possibility of presence of tropin alkaloids. If it is Convolvulaceae family, there is a possibility of presence of ergot alkaloids. Likewise, there are a number of other families which characteristically contain some or other chemical categories of compounds. So this is actually a significant mark. This can serve as a signal, or you can say this can also serve our purpose of selecting the plant material. And galenthamine is the success story. Galenthamine is actually used as a drug molecule for the treatment of Alzheimer. And this was selected from the genus Narcissus belonging to family Amaryllidaceae based on this chemotaxonomic approach. And this is one paper I got recently where they have tried to predict diterpene, various diterpenoids of the family Lamiaceae. Here a database is developed by the researchers in 2019 where a database of diterpenes is developed, and it is used to perform the chemotaxonomic analysis of the subfamily of Lamiaceae using molecular descriptors and self-organizing maps. And here what they have done is they have collected information on about 4,000 different diterpenes corresponding to about 6,000 botanical plant in which they occur, and they are which are distributed in 8 subfamilies, 66 genera and 640 different species, and about 5,000 different geographical locations. And they have developed a database which they say that it would be helpful in prediction of the localization of a diterpenoid, because they have gone for this many locations of this many geographical locations, and also in a subfamily. And this would assist in search of secondary metabolites with specific structural characteristics, like if this diterpenoids are of different chemical nature, like limonite diterpenoid, leptin diterpenoid, and many more. So this database would be able to say that if it is a leptin diterpenoid, this could be possibly present in family Verbinaceae kind of. Like, so this is how a database is available for chemotaxonomic approach also. So it makes us, so technology you can see has made our work very easy. Then this is example of ecological interaction. Next slide, please. So this is based on ecological interaction where one sees one observes the nature and goes for the selection of the plant material. Example is given over here is of Castrum diurnum and 3 other plant members of the family Solanaceae. And this here the researchers observed that cattle grazing animals were using these plants in specific conditions, and then when they screened this plant, they could see that the plants are reaching vitamin D3. Next, please. This is very interesting and very highly sought for approach, that is computational approach or network pharmacology, because here what is happening is we are having large number of databases which is actually helping us, and which, if not databases, we have the tools also which can help us in designing the our screening of the drug with plant material, that is medicinal plants. So this approach actually is a emerging tool, as an emerging essential tool for drug discovery, that is faster, cheaper than the traditional experiment based methods. Earlier what we used to do is we used to select the plant material, we used to screen the plant material phytochemically, and for then based on those results we will go for simultaneously or afterwards biological screening. But what here we are doing is we are having the plant material, and we know that from the various databases only, we know that what kind of active and inactive compounds are present in this plant material. If not active and inactive, we know that we have the literature regarding the phytochemical composition of the plant material. So what we do is we try to subject this phytochemical constituents to some in silico screening, and then we try to derive the possible targets, that if this a particular compound is going into the target and getting attached, so possibly it can be having this kind of effects. We can derive that theoretically, and then we can go, after getting this results, we can directly subject this results to the actual physical screening in the lab. So that saves our time and energy and cost also. So this approach actually is taking care of a reference of data set consisting of active and inactive compounds for a specific activity. We can derive quantitative structure activity relationship, that is QSR, and protein ligand docking methods can be used to analyze important structural features related to activity and develop the prediction model. As I told you, we can predict the possible target, and then from possible targets we can predict the possible disease also on which the plant material would be active. And further, we can also predict the absorption, distribution, metabolism, and excretion and toxicological properties also from this software, or from this with the use of this tool. And finally, we can illustrate the biological significance of an observed effect such that we can go for evaluation of this herbal remedy or medicinal plant, or also some individual compound, and we can even get the new ideas about the present therapeutic agent for the repurposing. That is, we can repurpose the use of that particular drug. Next, please. And this is actually, computational approach is also known as network pharmacology. This is the concept of network target which has been proposed to help design and predict the best possible treatments, as I told you, for the possibilities of higher biological activities. And this in silico simulations can be used to propose the protein ligand binding characteristics for molecular structures. Example, known constituents of the plants, whether if they perform well in their in silico predictions, can be directly used as a starting material for the experimental work. And the limitation of this method is the required availability of structurally and stereochemically well-defined compounds, and the approach inherent capability to find novel molecules constitute the limitations of these virtual screening studies. And here we have large number of databases available which would be helping us in getting this kind of work done. I can share the number of publications in this regard, wherein we have large number of databases available at our door to go for this kind of screening, like DIOS database, which is it's a collection of about 10,000 literature-derived compounds from the plants described by Pandanus Dioscorides in its Encyclopedia Materia Medica. Then there is a natural product database, which is a more general database that collects information about 13,000 natural products from the various research papers and databases. Then in CHM, there are about 11,000 compounds from traditional medicines. There is UNDP, which is Universal Natural Product Database, which is freely available database, and it can be downloaded also, and it contains about 2,900 compounds in its lab. So these are the examples of some databases, virtual libraries containing collections of natural products. Then these are again furthermore some databases like earlier I had said DNP. It is commercially available. Then it includes information on names and synonyms, physicochemical properties, molecular weight, pK, that is pharmacokinetic data, then solubility and spectroscopy data are there. So you can rely on this greatly because the information is very correct and precise, and from this you can design your further screening. It has information on biological sources also. Then the compounds are classified here in this database. This is a very systematic database, very useful one. It is classified into structural types with a total of over 10,050 classes, and it has information on UV spectra, biological sources, even toxicity data and dissociation constants are also given, and it is available both online as well as CD-ROM. So these are some examples of virtual databases which we can go for using our desired or drug molecule for drug discovery. Next, please. So this is one example which I have tried to quote here. This is example of one study which we conducted in our lab, wherein we have targeted in silico docking study of Cliteva nurvala bark constituents. Cliteva nurvala bark actually is reported in traditional literature for its utility in the treatment of benign prostate hyperplasia, which is a very common prostate disorder in male members above 50 years of age. So we selected the plant and we tried to get the literature on the chemical constituents present in the plant, and these chemical constituents were selected 20 chemical constituents were targeted for screening on the target protein pi beta reductase using Schrodinger software and using co-crystallized ligand finasteride, and with this co-crystal ligand we got score of 10 KCl per mole. Next, please. And these are the docking scores of the individual compounds which we screen for, which are reported in the Cliteva nurvala bark. This is the slide which shows docking score and ligand interaction of rutin. Rutin is a flavonoid, very popular one. It is having vitamin P-like property. It is useful in the treatment of capillary fragility. So this compound, it is a flavonoid. It contains two sugars, glucose and rhamnose, and this is a flavanol moiety which interacts with various amino acids like glutamine, glycine, tyrosine, etc. And the docking score we got was very high, that is minus 10.17. So it could be probably giving us very good activity when we actually go for the pharmacological or bioassay. Then this is the next one, quercetin. Next slide, please. This is quercetin. It is an glycon of rutin only, and here we have shown ligand interaction with the protein, that is pi beta reductase, and here docking score which we get is from minus 9.9. so it is again very high score which we are getting. So both rutin as well as quercetin. Rutin is a flavonoid which is derived from quercetin only by the attachment of the two sugars, and quercetin is again, both of them are giving very good activity. Here hydrophobic interaction was observed with tyrosine, tryptamine and isoleucine, methionine, leucine, tyrosine as well as tryptophan rather, and docking score was really good. Then third compound which gave very good score was retinoside, and ligand interaction with protein gave about minus 9.97 score, and this is again glycoside. So compounds with glycosyl nature, although I'm not, next slide please, I have not shown here the biological screening of this particular experiment. What we have done, we have, prepared extract and fractionated that extract into various fractions using from non-polar to polar solvent, and we have found that ethyl acetate extract containing this particular compounds like tretinosside and rutin and quercetin is showing very good activity in one more assay, that is brine shrimp assay. Artemia salina is the shrimp which is used here in this assay, and that has been shown us again. At this assay has confirmed our result of in silico study, wherein not the non-polar fractions like tetramether or chloroform extract, but the some semi-polar and polar extracts like ethyl acetate extract is showing very good mortality, or you can say IC50 with brine shrimp assay. I'm not shown that result here, but confirmation has been done further, and the process of doing this work on cell line is on. So this is how in silico method can guide us towards the success. That was my motto to show here this slide. Next, please. Now, once we do the selections, once we are over with the selection criteria, we are supposed to prepare the extracts and fractions for the phytochemical screening as well as biological screening, bioactivity screening, as I said, and biological screening. And various strategies employed are, we have to first extract, then that is the preparation of the crude extract is done, then separation is done using either liquid extraction or solid liquid extraction by simple solvents or by some other techniques. Then identification has to be done for the compounds which are present in the isolated from these fractions or separated fractions. Then analysis has to be done, and the bioactivity is done either at the end or simultaneously. If it is done simultaneously, it is known as bioactivity guided fractionation and extraction, and if it is done at the end, then it is known as biological screening only. So extraction can be done by conventional methods or by modern methods. Conventional methods take longer time, and their solvent required is more, and so these days we go for hyphenated techniques like microwave assisted extraction, pressurized liquid extraction, ultrasonication, use of flash chromatography, then ultra high performance liquid chromatography, medium pressure liquid chromatography, then high speed countercurrent chromatography, solid phase extract or supercritical food extraction method, well filtration method. These are all extraction techniques which are these days used for facilitating the speed as well as cost and energy. And also in some cases you can have better yield in terms of percentage contained or better yield in terms of quality also. So these are the advantages of hyphenated techniques. These I have tried to show here. Next slide, please. I hope I'm audible.
Yes, ma'am.
Yeah. So this slide shows methods which are conventional methods like maceration and percolation. This actually these days nobody is using, but if somebody if there is some non-availability of the hyphenated instruments, people can use this. And maceration is simply keeping the drug in contact with the solvent. This is actually a time-consuming method. It takes hours to few days to several weeks. And theoretically, if we see, if you want to go for maceration, you have to take drug to solvent ratio in the form in the ratio of 1:4:8:16. That is 1 gram of drug. If you are taking, you can take 4 ml of solvent or in that ratio only, and then keep that in contact with keep this drug in contact with the solvent for you can say minimum 3 days. That is ideally written in the textbooks, right? But it is of your the method can be optimized with our own choice and can be but one thing is there, you should always exhaustively extract the plant material by performing multiple cycles. Then this is percolation, which has been shown here. It is actually advantage over here. The advantage of this technique over maceration is it is a continuous process. Once you allow in the initial stage, you have to allow to macerate your drug with the solvent for a few hours, and then start collecting the extract from the bottom of the apparatus. Next is this is commonly used technique. It is a hot continuous extraction method. That is Soxhlet method. Only thing is you cannot use this for heat liable compound. It's a continuous extraction. You are using one single assembly wherein the drug is kept in the thimble. Your solvent is there at bottom, and heat is applied in the form of direct flame, or if it is non-inflammable solvent. And if it is inflammable solvent, you can always use water bath or heating mantle and start your extraction procedure. The solvent will vaporize. It will go and it will come in contact with the drug. These vapors will be collecting the solutes from the plant material, and it will come back because of the condenser. It will condense back in the flask which is at the bottom. So in a way, extraction and concentration of the the extract is going on simultaneously. So it's in some labs it is a matter of choice. Then these are some modern methods which I have tried to explain. One is countercurrent extraction method. Here wet plant material is pulverized to produce a slurry, and here what is done is it's actually chromatography method. There are columns in which a stationary phase is there, which is kept rotating constantly, and from the other end, liquid, that is mobile phase, is making entry into the system, and this liquid mobile phase will be taking along with it, because it is constantly running counter current. It is constantly running or passing through the stationary phases. It will be taking up solutes from the plant material, and you will be getting a rich extract at the end of the assembly. Then there is ultrasonication. This is a very simple instrument, ultrasound extraction. It is a simple water bath is there, which has got sonication facilities, and the procedure involves use of ultrasound with frequencies ranging from 20 kilohertz to 2000 kilohertz. This depends on the instrument you choose. And what happens is ultrasound in the solvent produces cavitation in the plant material, which accelerates dissolution and diffusion of the solvent into the plant material, and this improves the heat transfer as well as the extraction capacity or efficiency of the solvent. So it's a very good method which can be applicable for the extraction of thermolabile as well as unstable compounds. So these are some simple hyphenated techniques, that is technological interventions you can say, which can be used for the enhancement of the yield, quality of the product, reduction of the time, energy and cost. Then this is a very highly valued or highly used method, supercritical fluid extraction method, where the supercritical carbon dioxide is very widely used, because and that is used at a lower critical temperature and pressure. And the critical temperature for the supercritical carbon dioxide is 31 degrees centigrade, because at this temperature its selectivity, inertness is very high. It is also very low cost solvent. It is non-toxic, and it has capability to interact with the thermally liable compounds. And so this method is it's a method of choice for extraction of non-polar natural products such as fatty acids, lipids, and volatile oils. And when it is used for compounds like secondary metabolites of our desire, like alkaloids, glycosides, and some other compounds, then we can always use a modifier or can be a solvent like ethyl acetate, methanol, which will be enhancing the solvating properties of this supercritical carbon dioxide. So in a way, by using modifier, you can have the selection of this method for section of alkaloids, glycoside kind of compounds also. So this is a graphical presentation of this kind of the supercritical extraction method. You can see that there is a chamber on the left side on the screen, where there is a carbon dioxide supply is there. There is a pump in between which is pressurizing that entry of this carbon dioxide into the extraction vessel, which is there up here. And if you want to add co-solvent or a modifier, there is a reservoir for co-solvent also, which will be pumped into this mixture, where both of them will be getting mixed. That's carbon dioxide as well as co-solvent will be mixed, and through heat exchanger, that these both will be getting entry to the extraction vessel where our material has been kept, and this solvent will be carrying along with it under the critical temperature and pressure condition. It will be going to through this pipe. It will be going to the collection unit, and from that you can drain the extract out from the lower point, and the extract, the solvent can be, for the solvent also there is one more vent which can be recycled, collected and recycled in the assembly. So this is a supercritical fluid extraction unit. Next one is accelerated solvent extraction method. This is also known as pressurized liquid extraction method, wherein we are using, we can use here, in fact in our lab we are using, for this particular method we are using autoclave. We do this method at the temperature about 110 to 115 degrees centigrade and at pressure of about 215 psi, right? So this can be done using simple autoclave also. You have to seal your container in which you have kept your solvent as well as drug. You have, and this method can be completed within short time. You can get very good quality of extract, right? About 5 to 10 minutes your extraction procedure would be completed. So this is a very good method of choice for faster extraction, and this results in high solubility and high diffusion rate. The conditions are that the solvent will be having high power under that condition, and so the solids will be taken up very easily from the solvent, by the solvent, and you will be getting this extract of high quality within shorter time. But there are disadvantages also, sometimes if you are not properly optimizing your conditions, the content of the flask get expelled. But that is only in case of you are using autoclave. If you are using systematically the instrument itself, there are no such possibilities which can happen. Then this is next is extraction assisted with microwave or infrared. Here microwave is used to generate heat by interacting with polar compounds such as water or some organic components in the plant. Matrix can be extracted because of the ionic condition and dipolar rotation mechanisms. And here the heat and mass transfer are in the same direction, and so this gives better yield as well as faster extraction. There are two types of microwave assisted extraction method. One is solvent free extraction method for volatile compounds, where you are using the combination of microwave heating and dry distillation, and this method is performed at atmospheric pressure without adding any solvent or water to get volatile compounds in a single stage. And there is one more method where solvent is used in microwave, and this is for basically non-volatile compounds, and the advantages are higher yield, thermal degradation is minimized, and it is considered as green technology because we are not using any organic solvent. Then this is enzyme assisted extraction method, where we are using enzymes. These enzymes will be coming in contact with the polysaccharides or proteins, and they will be resulting in the coagulation of this coagulation as well as denaturation of proteins at high temperature. So you can remove these things, and whatever is left can be used for your desired product. Like if you do hydrolysis, if you want to go for isolation of glycosides, always glycosides are present in the plant material in the along with the enzymes. Like in a tissue, there is a presence of a tissue is made up of multiple cells. Some of the cells will be containing glycosides and some of the adjacent cells will be containing enzymes. Now what happens when you try to give physical damage to this tissue? Enzyme will come in contact with the glycosides, and glycosides will get easily hydrolyzed, and this hydrolysis will lead to formation of small compounds, that is aglycons, which can be easily isolated. So what can be done is we can use this property of hydrolytic action of enzymes on this kind of compounds to get smaller molecules which can be easily isolated and characterized also. So this is one more method which is you can say technological intervention in getting the desired compounds. Now after getting the compounds, we have to actually go for, after getting the extracts and fractions, we have to actually go for separation. Now extract and fractions, you all know they are mixtures. So how to separate the compounds from these mixtures? And for separation, there are multiple methods which are available. In earlier days, we used to do preparative TLC or paper chromatography in a preparative manner. But these days, and then at the most, we used to do column chromatography, which was actually very widely used for the separation of natural products from any, and because it was very simple, a column of your size, your capacity can be used, and the adsorbent, which was actually simply silica gel or macroporous resins at the most. They were of low cost, but with the use of these things like column chromatography or preparative TLC, you can get the separation of your compound from the extracts or fractions, right? But with the advent of, in this case is also, not only silica, we can now have in the modern era, in the recent years, use of alumina has increased, use of polyamide for flavonoids, use of various, you can say dextran modification of polysaccharides, that is Cephadix-like compounds. Various categories of Cephadix are available for using it for the column chromatography also. And one more thing, when you do this column chromatography, here also several modifications have come, like vacuum column chromatography. If you apply vacuum to the column, your separation is going to be on the better side. So that is a technological intervention. If you use pressure for the column, that has been used currently in the flash chromatography technique, which is recently only coming in the light, like last 10-15 years we are listening about flash chromatography. This is using simple it is a column chromatography only, but here the columns are pre-packed, that is with pre-packed means the adsorbent is pre-packed in the column. We are making the entry of the mobile phase through systematic pressure pump in a controlled flow rate manner, and we are eluting that, and we can also detect the elutes. Whatever we get eluted at the end, we can directly sub it to the detector also. So these are the things which are available these days with the for the as a modification for column chromatography. Next, please. Then there is separation based on partition coefficient, partition between the, and this actually is taken care by liquid-liquid extraction principle. When there are two liquids, this is the principle which acts behind that. And this principle is used in counter current chromatography, which I said here, liquid as well as, stationary as well as mobile phase, both of them are liquid in nature. Only difference is liquid stationary phase is holded by gravity or centrifugal force, through which the current of liquid mobile phase passes, and taking with it goes to the collection unit or to the detector. And here these days, high speed counter current chromatography as well as centrifugal partition chromatography are widely used for isolation and separation of variety of molecules. This is the next slide, please. This is the pictorial presentation of the same. You can see stationary phase is stagnant by centrifugal force or gravitational force, and mobile phase is moving. And this is the entry. With pressure it goes inside, and from the other end it comes out with the solute from the plant matrix. Next, please. Then separation technique by molecular size. Here membrane filtration of your choice are used. Microfiltration, ultrafiltration, nanofiltration based on pore size can be used. And this is a powerful technique for concentration, clarification, and removal of impurities from the fractions or extracts which you are getting. Then there is next gel filtration chromatography, or that is also known as gel permeation chromatography or size exclusion chromatography. These are here what is happening is small molecules have longer retention time because it is a gel. So smaller molecules come out earlier than the larger molecules. This is the graphical presentation of that. You can see I've shown that this is a gel permeation chromatography graphical presentation. Small molecules are penetrating easily, and they're coming first, and separation is obtained. Then this is actually very recent entry. Technology, how it can help us. You can see here a very interesting one also, molecular imprinted polymer. It is an artificial acceptor made by imprinting molecules of a template in a polymer matrix, followed by removing the template molecules via thorough washing to give the permanent template grooves. And this technology offers an effective, efficient separation and enrichment of specific analytes from complicated matrices, and it has been used for separation in recent years due to its high selectivity, good stability, and simple preparation. It is actually used to remove chlorophyll from the leaf extract, which is always rich in chlorophyll, or sometimes for the separation of a particular compound. like many contemporary complementary cavity, how it can be done. Many complementary cavities with a memory of size, shape, and functional groups of the template molecules are generated when a template molecule, and when these template molecules, how they are generated, first they are placed, and then when the template molecule is removed, this molecular imprinted polymer is ready for use. This shows favored affinity for to the template molecule compared to the other molecules. And this property is basic driving force for such diverse application of this technique. And this is increasingly employed in a wide scope of applications such as chromatography, sample pre-treatment, purification, and drug discovery, etc. Next, please. Hello. Am I audible?
Yes, ma'am.
Okay. Archana madam called me.
No, I didn't call, ma'am.
I got the call from you. Anyways, I'm audible, clearly audible. So
yes,
this
yes.
Yeah. This technique, I'll be now giving the example. This technique has been widely used in the separation of natural products or a solid phase extraction sorbent. Again, solid phase extraction sorbents are available ready-made, wherein you can use simply them. You can simply use the sorbents and pass your fraction or extract through this with a selective solvent, and you will get the separation through this solid phase sorbents also. So, and these solvents can be co-used with multi template molecularly imprinted polymers. And the example here given is, I have given is of separation of denchinin from the water extract of Panax ginseng. And this separation is done by using DL-tyrosine and phenylpyruvic acid as a template molecule. Another example is a preparative separation method to separate solanisol from tobacco leaves has been given using this molecular imprinted technique. And here what they have done is MIP is prepared by methyl methacrylate as a monomer and solanisol as a template molecule, and ethylene glycol dimethyl acrylate as a cross linker. And this was prepared by suspension method. I'll not go into the detail of this because it's a long method. Then next is, after you get the separation, now we see, we can say that we have either enriched fractions which are which can be subjected to fingerprint profiling, or directly different compounds are there which can be subjected to structural illustration. Earlier days, structure fingerprinting for the fractions, enriched fractions, was done using TLC or HPLC, right? And structural illustration was done using chemical reactions. They will start do the fragmentation using variety of chemical reactions like reduction, oxidation, and cleavage of the main molecule into small molecules, and that is how they used to derive entire structure of the molecule. But these days we have spectroscopic techniques, and spectroscopic technique also, when we combine this with hyphenated techniques like HPLC or GC, or HPLC or GC, then it becomes a combination of techniques. So that is hyphenated technique. So hyphenated techniques are very useful these days, and every lab is using this kind of techniques to go for analysis as well as identification of drug molecules. It's a technique. It's a combination of or coupling of two analytical techniques with the help of proper interface. It is developed by coupling of separation technique and detection technique. I am telling you, GC, HPLC combined with spectroscopy, either UV, IR, NMR, mass, any, right? So it's a combination of separation technique and detection technique. And term hyphenation was first used by Hirschfeld in 1880. And these are some next, please. These are some techniques which I have tried to enlist, like high performance liquid chromatography, diode array detection or UV detection, then high performance thin layer chromatography, UV detection or mass detection. It is NCHROM is giving this instrument with where HPTLC is provided with interface and mass apparatus, the instrument, where directly the resolved spot would be taken up by the interface and subjected to detection by to the mass detector, and you will get the identification of that particular spot. Then GC along with mass spectroscopy, GCMS, then GCMS-MS, GCNMR, GCAES, that is atomic emission spectroscopy, GCIR also is coming in. So these all hyphenated instruments are available which can be used for analysis as well as detection of, identification of our natural product. Then there are a few more of them. Capillary electrophoresis, diode array detection. This particular technique is specifically used for DNA fingerprinting of the raw material, which I'll be talking about later on in a brief. Then HPLC mass, HPLC NMR. These are all techniques which are used in metabolomics, genomics, and proteomics, and even in fingerprinting as well as chemometric analysis of plant drugs. And this does not always have to be between two techniques. That is coupling of separation or detection techniques. There could be coupling of separation, detection, separation, and the other way. And we can have very good resolution of this extracts or fractions or even compounds. And these techniques are simply coupled with computational algorithms to get fast, accurate analysis, better reproducibility, reduction of contamination, and separation and quantification at the same time. Because unless this data generated through these techniques are very complex, and so we require computational algorithms, as discussed earlier by the speaker also. They are highly used for bioinformatics and chemometric types of analysis, and then only we can successfully go for analysis of or use of these hyphenated techniques for our purpose of medicinal plant research. This is one paper which is use of simple HP, you can say not simple TLC, HPTLC, wherein in our lab we have tried to quantify simultaneously 3 compounds from a methanolic extract of Soralia corifolia seeds. Bavacin is a flavonoid, bakuchiol is a maroterpenoid, and soralain is a coumarin, furanocoumarin. So these 3 compounds are belonging to 3 different categories, and that is bavacin is a flavonoid, bakuchiol is a maroterpenoid, and soralain is a furanocoumarin. So all these 3 compounds we tried to quantify using simultaneous HPLC method, and a mobile phase was developed, a scanning wavelength was optimized, and we published this paper recently in Journal of Paper Chromatography. You can see here, this is bavacin, this is soralain, and the spot which is resolving in higher RF is bakuchiol. All 3, I'll repeat, all 3 are of different chemical nature, and we established a method which is simple, precise, and reproducible, and it can be used for the quantification of all the 3 at a time, and this would serve as a purpose of standardization of the extract of Soralia, because standardization is very much required in these days. With those who are preparing herbal formulation, they should be using standardized extract. So if a person is having Soralia extract, he can use this method, and he can standardize his extract using this method for the content of these 3 molecules. This is next a publication from my lab. Again, a development and validation of rapid LC-MS/MS method for simultaneous determination of camphorol and quercetin. This is LC-MS/MS method, where with the use of this method, we qualify the extract for the presence of camphorol and quercetin, and also quantified them in the extract which we had used. And here also we had optimized the column condition, we had optimized the mobile phase, we had optimized the detection wavelength, detection for the mass, and we could successfully quantify the content as well as qualify the presence of, we could identify these 2 compounds in the extract. See, advantage of hyphenated technique, that is combination technique, is you can identify also from the mass spectra. HPLC would be giving you the fingerprint, that is peak, and this peak will be whatever is getting eluted first will be going to the detector first, and detector will be detecting the identity of that particular compound, and you will be able to identify or know that this compound is of this particular nature. Like in our case, camphorol was resolved first, so the peak which was resolving at about 3.2 minute was of camphorol, and which was matched with standard also, and we could quantify successfully these 2 compounds in the extract. Next is biochemometric use of biochemometric analysis. Use of biochemometric metrics in the plant drug analysis. This is the example I've taken from some study which has been reported by a Chinese author, wherein I'll show just that. Next slide, please. Here, what they have done is they have taken one extract of a plant, traditional Chinese medicine. They have done the literature survey and they have enlisted the number of compounds present in the plant extract, and they have categorized these in the 3 different families. Here there are 3 colors which are visible in the first herbal extract label. જે કર્યું છે ત્યાં 3 color દેખાય છે, green colored compounds, red colored compounds and purple colored compounds. So chemical family based classification was done by the authors. Then it was subjected to preparative HPLC. Preparative HPLC is also advantageous to go for because it gives us fraction. You can collect the fractions which are getting diluted in the HPLC, and then that fractions can be subjected to the mass or any other detector which will be giving us identification parameters. So here they have used preparative HPLC to get the separation. They collected these fractions in the test tubes or the collection units, and then what they have done is they have rebuilt the family chemical compounds by adding. Fortification is done here. You can see in the first graph there is lesser concentration of the compounds 4, 5, 6, but in the library reconstruction there is a higher concentration, right? And then this reconstructed extract is subjected to the bioactivity distribution map. That is, bioactivity is done, subjected to the evaluation of the bioactivity. And this graph shows that compounds which are represented here in green color, they have got higher bioactivity. So you have now the classification of these compounds into bioactive compound and non-bioactive compound. Now what they have done in the next step is they have done multi-component interaction between intra-family and inter-family. એટલે green માં combination કર્યું, પહેલા separate કરીને activity જોઈ, પછી combination માં activity જોઈ, પછી inter-family combination જોયું, green red અને green purple, and they have tried to get the idea about several algorithms. They subjected this data to several processing of software for algorithm calculation. Computational algorithms was performed, and they derived additive, antagonism and no interaction kind of separate idea about each of them. And then whatever was giving additive action were subjected to in silico study for the through molecular docking study, and finally they get the idea about the active fraction, additive fraction, and they derived the biologically active fraction by actual bioassay. So that was the use of biochemometric or chemoinformatics, or you can say computational algorithm along with semi-preparative or preparative HPLC and mass, right? So hypothetic technique coupled with computational algorithm was studied here. Now this is next is use of these techniques in the metabolomics. I'll not go into detail of this this particular section because I think earlier speaker had given some information about this. Metabolomics is actually a very useful technique. It can be performed using HPLC, MS, HPLC, NMR, GC, MS, likewise. And this data, whatever has been generated, it is to be coupled with multivariate software to get the idea about the results. Here, this is used for authentication of this plant material, and we get idea about the broad range of secondary metabolites present in the drug. Not only metabolites present in the drug, this metabolomics can be studied for the pharmacokinetics, pharmacodynamics parameters also. That is what is happening in the body once the drugs are consumed. So this is metabolomics, and this is the example of the work done in my lab. Using this approach, using GC-MS technique, we have screened Juniperus indica samples collected from different valleys of Uttarakhand. We targeted 13 samples collected from Niti Valley, Byas Valley, Dharma Valley and Johor Valley of Uttarakhand, and subjected to these samples we isolated volatile oil from these samples and subjected these samples to the GC-MS study. And these are some the graphs of this GC and MS. You can see the graphs of these samples, and these are the mass spectra of the major compounds: sabenin, philandrin, and carine. Go to the next third slide, where one can see graphs of mass spectra of sabenine, philandrine, and carine. These are the compounds which are present predominantly in these volatile oils. And on the basis of this, we can conclude certain geographical pattern, distribution of certain components depending on the geographical pattern. You can see here, this is the table. Next slide, please, which shows the table. Please go to the slide which shows the table. Is it shared? Archana, madam?
Yes, we can see, ma'am.
Yes,
So
ma'am.
the table shows that the sabenine, first compound, is present in 1 to 6 samples, and these samples are from Niti Valley and Vyas Valley. And these samples are of berries as well as whole twigs. They show this compound in predominance, while those plants which were collected from Zohar Valley and other valley are showing less of this compound. You can see that a clear difference is there. So if one wants to go for having, if one wants to have more of this compound, that is sabenine or thujane, which is a monoterpene, bicyclic monoterpene, major bicyclic monoterpene present in this volatile oil, one should be selecting this plant from the Niti Valley or Vyas Valley, right? And if we see the content of other compound, that is carine, again the distribution pattern is following the same, that is pattern of fusion only. Carine is the 10th compound, which it again is present in more in the samples of Niti Valley. So this is how we can locate, we can try to go for the better crop to get better kind of compound. Even terpenine is present in more in amount in the samples of Niti Valley. So this is how we can, with the use of this technique, we can verify the presence of desired compound, and we can target our cultivation as well as biological activity to get a better crop. This is cluster analysis, which says same whatever I told you, that the predominance of sabenine as well as carine was found to be there in Niti Valley and Vyas Valley, while Dharma Valley and Johar Valley samples were not containing that much amount of these two bicyclic monoterpenes. So this is how we have used this technique for locating the good samples of Juniperus. Then next is proteomics. It is an approach to innovative drug discovery from natural source to illustrate protein expression, protein function, metabolic and biosynthetic pathways. This has been discussed by earlier speakers, so I'll not be going into the detail. Next, please. Genomics. It's the use of this hyperrated techniques, specifically electrophoresis is used in this to go for a DNA fingerprinting of the botanical material. If we are conventionally, if we are trying to identify, we will prepare herbarium, and we will compare our sample with the herbarium, or at the most we'll go for morphological identification or microscopical identification, right? But DNA barcoding also is a better technique for the authentication of this plant material, and that can be done using electrophoresis technique. Next, please. Then last would be biotechnology. Production of biopharmaceuticals. Again, this has been discussed by earlier speaker, Dr. Sharma. He gave very good examples of the number of techniques to improve agricultural crops of rice and banana. But here I am trying to quote biopharmaceuticals because I am from the pharmacy background. I deal with drugs or source nature as a source of drugs. So here biopharmaceuticals, with the use of biotechnological tools, we can go for production of biopharmaceuticals. The concept of using plants to produce recombinant pharmaceutical proteins is referred as plant molecular farming, PMF, or farming, that is PHARMIG, PMP. It is not new. Human growth hormone, initially produced in tobacco and sunflower in 1986, was first plant-derived recombinant therapeutic protein. And since 1994, about more than 100 pharmaceutical proteins have been characterized and expressed and characterized totally. And by 2011, more than 20 phytopharmaceuticals are there in the market. They were subjected to preclinical or clinical trials, and successfully some of them are there in the market also. And it represents a promising system for production of human pharmaceutical proteins on large scale at a low cost. It also has the capacity to produce post-translation modifications. The use of plants also eliminates contamination of the therapeutic drug with animal pathogens. So increased safety is there. In general, the cost of PMF derived product is only 0.1% of mammalian cell culture systems and 2 to 10% of microbial systems. So always, plant cells are better option than the animal cells or microbial cells. And many production challenges are there, but it could be, I think, the hurdles would be overcome by in the coming years. Next, these are some examples. This VEGF is produced by one of this researcher, and this human VEGF is produced from the barley grain. It has been commercialized for the research purpose, and this plant-expressed human VEGF is used in the treatment of thinning hair, which is a problem in the majority of the male members. So this is one of the examples which I wanted to share, and these are a number of other examples wherein this biopharmaceuticals are in use for the treatment of Bucher's disease. Then an chimeric secretory IgG or IgA antibody is effective against surface antigen of Streptococcus mutans, which has been prepared from the tobacco. It has been expressed in the tobacco, and this has been demonstrated to be effective in against dental caries. Then transgenic potatoes also can produce antigens of enterotoxogenic E. coli, which is a heat-liable enterotoxin BU subunit, and this is effective in immunizing against viruses and bacteria that cause diarrhea. Then edible vaccines are under development for rabies, foot and mouth diseases, for specifically for veterinary purpose, even cholera and autoimmune diseases. Then transgenic lupin and lettuce plants can express hepatitis B surface antigen, which is a very acute problem. And there is an example of tobacco leaves giving us ZMapp. It is used to combat 2014 Ebola virus outbreak in Africa. So these are some examples of biopharmaceuticals available, success stories already in the market. And lastly, I would like to summarize my talk that innovative selection method with the use of software, digital libraries, then innovative extraction methods like use of supercritical food extraction method, accelerated solvent extraction method, molecular distillation, and all. Many more things can be, and even separation technologies can enhance our capacity to get better fractions or directly a single compound. Then this can be subjected to in silico studies prior to the actual studies for biological assays. And by using bioinformatics and multivariate analysis, we can have the very good analysis or data which can be further subjected. And omic technologies can be always used for the better quality yield, faster process, and many more. So thank you all for the patient listening. In between, I troubled you a lot, but then
No, ma'am, it's fine.
technological problem again. I was talking about technology. Technology only put many hurdles in my talk.
Never mind. Sanjukta?
So thank you so much.
Thank you, ma'am.
Yes, ma'am. Yes, ma'am. Thank you, ma'am. Thank you for such an informative session. With this, we come to the end of the session. We'll be sharing the feedback form link in the chat box, and your e-certificates will be generated thereafter. On behalf of Department of Botany, Bioinformatics, and Climate Change Impacts Management, Gujarat University, I once again thank you all for your participation.
Thank you.
Thank you so much.
Thank you.